As we said, we're talking about today about engaging with end users to explore the acceptability of technology still in development. And if I can get the slides to move, there we go. So we know that a lot of new technologies for health don't end up being implemented at all, or they're abandoned after a short period of time because they're not seen as fit for purpose by the people that are meant to be using them. But we also understand that incorporating end user needs into the technology design can help mitigate these types of barriers. But the consequences of technologies are not always known, and that's specifically true when they're still under development. And also just asking end users for what they want can end up producing a type of wish list of things that are not actually technologically possible, leading to frustration for both producers and the end users themselves. So questions still remain about how and when we can engage end users in technologies under development. So as you know, we'll be presenting two studies. The first of these is the PRECISE study, which I'll be presenting now, and that's on stakeholder input into the development of point-of-care tests. And then Jonathan will present the new COTIANA study, which is around understanding producer opinions of shifting tobacco farming for consumption, like smoking, to modified tobacco for the production of high-value molecules to be inputted into health products and other types of products as well. So to move on to the PRECISE study, as we said, this is around development and evaluation of point-of-care tests for the detection of multiple STIs and antimicrobial resistance. And this was to be held in collaboration with experts, patients, and the public. The study ran between 2015 and 2018, was hosted by St. George's University of London and funded by the NIHR Invention for Innovation program. And we did this in collaboration with a company called Binks Health, who's producing the tests. So there you can see the test that was under development. That's the reader, and they come with cartridges. And the idea overall was to design target diagnostic assays and then transfer those onto cartridges for 4 common STIs, and that's chlamydia, gonorrhea, mycoplasma, and trichomoniasis, as well as antimicrobial resistance detection for gonorrhea and mycoplasma infections. So the idea is that we couldn't get all 6 of these assays onto one cartridge. So how do you design it? And the decision for multiplex designing was based on both technological capacity as well as user preferences. So to meet that last aim, I designed a social science study to describe both the current care pathways in sexual health clinics across different settings in the UK, and then understand these ideal configurations for the tests. So that included around knowing whether or not we should have and how we should have multiplexing on infection detection, so which infections to put on what cartridge, whether or not patients and clinicians would find waiting for test results for about 30 minutes to be acceptable, as that is longer than would normally be the case. And for antimicrobial resistance detection in gonorrhea and mycoplasma, understanding whether or not this should be a standalone antimicrobial resistance test, or if it should be included with infection detection. And that's quite important because we need to know about the acceptability of, for example, waiting for results of an antimicrobial resistance test for those positive patients, as this would allow them to have their treatment be guided. So in order to meet these aims, I designed a study that was both a qualitative study and included some public engagement. In the qualitative study, it was informal interviews with clinicians in purposefully chosen sexual health clinics and individual in-depth interviews with patients in each of those clinics, and then holding a public advisory group consultation, so a series of meetings over about a year. To analyze these, both for qualitative study and the public advisory group, it was based on a content-based analysis, identifying common themes, using a common mixed inductive-deductive approach. Our PAG meeting notes were coded by hand, and the patient interviews were coded in vivo. And we analyzed both of these data sets separately and then compared and contrasted themes in a combined analysis. So I'll present some of the data here and point you to where you can get the rest of it if you're interested. So a qualitative study was held in 6 clinics across the UK. You can see the locations there. And these clinics were chosen to have diversity of implementation pathways for diagnosis and treatment. Some of them included new types of technologies, new types of testing methods or patient recall methods, and others were much more traditional types of care pathways. So I held informal interviews with clinicians in each of these clinics, and those were the clinicians that would be actually formulating the care pathways for these infections and their treatments. And then held one-on-one interviews with patients that were purposefully sampled based on age, gender, and sexual behavior, as those would be the areas where they would be guided on different types of care pathways, and aimed for about 9 patients per clinic. So in our clinician interviews, we discussed current care pathways. You can see a example of one of those there. You're not meant to be able to understand it, but I will go through that in a little bit more detail later. And by understanding those current care pathways, we then could move on to discussion around the potential use of the new tests, including the new clinical care pathways that would be associated with the different types of iterations of the test itself. And then we discussed, of course, the clinician preferences for point of care test cartridge configuration, not only for the product as it was looked at now, but at the time rather, but for the future as well. So just to go through this a little bit briefly, I don't know if you can see my mouse here, but effectively what you would do coming into a traditional care pathway, and this is a very simplified one, is that you would, after triage, you would then be brought in for a consultation as a symptomatic patient. There, the clinician would collect samples, most likely would be collected by the clinician, and then take sexual history would be taken, as well as any symptoms. So based on that, a subset of these patients are going to be presumptively treated for infections, but all of them will have their samples sent to a laboratory in about a week. The results come back to clinic, and then the result is sent to the patient from the clinic, either via text message or a phone call. And those patients that weren't already treated presumptively in clinic, but were found to be positive in the laboratory test, would be asked to reattend and then given treatment then and there. So unfortunately, the presumptive treatment algorithm is not terribly accurate. So there are quite a few people that might have not been treated correctly or were given antibiotics when they didn't need it. And then there are a subset, of course, that will be found positive later on that would have to return to the clinic. So the point of care test tries to reimagine and improve on all of that. And how the clinicians spoke about that and how the test would be used falls into 2 main categories. One would be a type of sample first pathway. So that would put patients into the waiting room after they collected their own samples, and they would then wait in the waiting room for both the test results. When the test results were ready, they'd be called in for their consultation, and they would have their results ready at the consultation time. The other way of doing this that was discussed was to keep existing care pathways, so ask patients to come in for their consultation, go through the normal care pathway as described on the previous slide, but then ask patients to go back into the waiting room to wait for their results, or to use a queuing management system to recall patients when the results are ready, which is an interesting kind of glimpse at how some technologies will necessitate the use of other new technologies. And there are all sorts of different iterations within that, but I won't go into those in too much detail. So these interviews allowed us to construct the patient topic guide, where we first asked patients to describe their most recent clinical visits so that we could get an understanding of their experience of the care pathway, and it could be quite fresh in their mind what they just experienced in the clinic, and whether or not they experienced any alternative care pathways in different clinics in the past. We then went through and discussed the general impressions of the point of care tests and gave increasing information about what the new point of care tests could mean for them in terms of direct changes within the clinical care pathway, such as waiting 30 minutes or more for their results, receiving results for some of their infections at one time, and then waiting for others. The possibility, again, of this reflex testing, so waiting another 30 minutes for their antimicrobial resistance test results so that they would have to wait for their treatment, and the potential for increases in self-sampling. So overall, we interviewed 63 patients in these 6 clinics, and I've got a link there on the article that is reporting our full results of the qualitative study for those of you that are interested. But just to go through some of these really briefly to give you an understanding of the type of data that we were able to gather from these exercises. What we found was that the acceptability of a general point of care test was very high. So that could be thought of as expected because it was you'll be able to get your results a bit quicker type of scenario. But the acceptance became a lot more situational as the details of the implementation were discussed. So patients described the circumstances where they would be more willing to wait for the results at the point of care and why. So this patient is a really good example of this. And he says that, I think it all comes from gauging how much at risk I am at the time as to how much amount of time that I'm willing to put in to getting tested and getting it sorted. In our public advisory group, we had open enrollment to all members of the public, and we held multiple meetings to discuss first the current care pathways, point of care tests, and antimicrobial resistance to give everybody a sort of level playing field of knowledge and discuss the intricacies of these types of things before we discuss the actual point of care tests that were under development and what their implementation might entail. We had a final meeting to assess their opinions of the point of care tests, and that was a discussion that was both prior to and after the interview results discussion, and that was done so that we could understand and compare the results from the public advisory group versus the qualitative interviews. So we had about 4 people from the second meeting onwards in our advisory group. We held 4 meetings between March and November in 2016. What we found was strikingly similar in many ways to our patient interviews, and that was that the possibility of the tests were seen as positive, and that acceptability of new care pathways and waiting for results would be mainly based around patients' reasons for attending. So that harkens back to this idea of why I've attended, and if I feel at risk, then I might be more willing to spend quite a bit of time getting it sorted out on the day. They talked about the advantages of the tests, as they might be able to provide better patient pathways and could increase patient choice for ways to test, and could also, crucially, encourage high-risk patients to test or screen more frequently because of these factors. Some of our key findings in our combined analysis in the patient interviews were around, we looked at recommendations for implementation where patients' expectations, again, were based for the future visits, were based on their past experiences, and that providing patients with information on the changes to clinical pathways would be important for them to have at or before their attendance, ideally, so that they could plan how much time it would take if those timelines of how much time they normally spend in clinic were to be changing. They really focused on the acceptability of the proposed point of care test pathways in the interviews, even when discussing recommendations for improvement or implementation of the tests. Whereas in the public advisory group, the recommendations were to encourage, to use the test to encourage attendance, but to make sure that point of care tests don't shift waiting times from home, as is current, to the clinic, and to advertise the speed of results delivery as well as the benefits to antimicrobial resistance testing to encourage the test's acceptability. And this group really focused on the potential for advocacy that these tests could potentially provide. So what happened with the actual test development? Well, in the short term, the decision on cartridge configuration was actually made prior to our final study findings. And the decision was to triplex the infection detection test and multiplex both the gonorrhea and mycoplasma antimicrobial resistance tests. We found that market research outside the UK drove focus on the inclusion of trichomoniasis in multiplex cartridge design, and that's because of the high incidence of that infection in the United States, which holds quite a bit of the global healthcare market. In the longer term, the 4-infection multiplex test and the gonorrhea and gonorrhea antimicrobial resistance tests both have been taken forward as product lines. They would have been actually launched already, both in the US and in Europe, but they have been delayed for a shift in focus by the company to COVID diagnostics. So strengths and limitations of this approach, just a few of them, and I hope that we can discuss more in the time for questions after. So our limitations are that in-depth qualitative research is really slow, very, very slow compared to market surveys and understanding the pressures and timelines from companies and, of course, the company's investors. And that may necessitate quicker decisions, at least in the short term, around product design lock. And the opinions and patients and public advisory group members remain hypothetical. So we know, of course, that people do not always do what they say that they're going to. So, quote unquote, more research is needed to show what happens in actual practice. And of course, our public advisory group member opinions are unlikely to be generalizable, obviously not on the population level statistically, but also not theoretically in a qualitative sense. The strength of our approach is that we were able to gain multiple perspectives through critical engagement of all of our stakeholders, so that's clinicians, patients, as well as members of the public, and that providing the context of these existing pathways helped to situate the hypothetical new pathways and uncover potential consequences for implementation. And the clinical accounts of potential new pathways helped us to determine how the new tests are likely to be used in practice. So it was really allowing us to make concrete these hypothetical situations. And our findings do go beyond development concerns to assisting with implementations. And although the timelines might be too slow to inform on immediate product development, actually the developers can use this knowledge in future product iterations, as was shown in the previous slide.
So yeah, I was along with Sebastian, we were part of the New Cotiana project, which I joined in spring 2018, and we've been running under the banner of Growing Health for a little while. And this is all about stakeholder attitudes to something called molecular farming with tobacco. So what is plant molecular farming? It's basically turning plants into bioreactors, which is how we currently develop or produce high-value medical molecules for vaccines and all kinds of things. And there's to give you a concrete example, this is VRC01. It's a broadly neutralizing antibody, which can be used to fight the HIV infection. And colleagues at St. George's, where we used to be based, had actually produced this in tobacco. And this is my tobacco plant Smoky, which is doing pretty well in the Devon climate somehow. And there's a few reasons why I'd want to produce these things in plants rather than a bioreactor. Lower costs, potentially. It's kind of infinitely scalable because you can just keep producing more plants. Bioreactors tend to be quite expensive, the stainless steel kind. You can localize your drug production as well to local needs, and there's low risk of human pathogenicity. So certain types of bioreactor can actually be infected with communicable diseases, so CHO cells, I think they're called. We also have cultivators all around the world who can grow things, particularly in the case of tobacco, which is kind of a model crop for this kind of research. So yeah, those are the reasons you might want to do it in plants. What makes this possible is a suite of technologies called new plant breeding techniques, which are pretty badly named because they're not all new and they're not all plant breeding techniques. The most transformative of which are site-directed nuclease. These are like things like CRISPR, which you might have heard of, which will certainly have medical applications as well. Zinc finger nuclease, talons. We also have agroinfiltration, which is where you kind of infect the plant with a bacterium and deliver a load of recombinant DNA and get the plant to produce what you want that way. You can also graft, which is a really old technique of taking the rootstock of one plant and grafting on another. So you could have a modified rootstock and a non-modified graft. What this allows you to do is something called cisgenesis, which is different to the transgenics of old genetic modification, or at least biologists like to say that this is different. And it means you can use you're only using native DNA. You're not introducing anything foreign. So this could change things legally from the perspective of genetic modification and potentially public acceptance, which is kind of what we're looking at here. These are the things called gene editing. You'll probably hear that term more and more in the future. So the New Cotiana project really set out to develop tobacco specifically as a molecular farming platform, create new varieties for open field production, and to explore the potential risks and benefits of doing that and find ways of scaling it up. And this was our bit as the social science wing of the project. So what we did was interview key stakeholders in tobacco producing regions of Europe. And there are tobacco producing regions of Europe, mostly Spain, Italy, and Greece, but a little bit in Bulgaria as well. So we spoke to 15 tobacco producers, farmers. We spoke to 7 technicians, 1 researcher and 1 policy expert. Here I've highlighted the regions in Italy and Spain where we were based. And in those places, we had a peer interviewer at each location to help us organize and then translate during the interview process, because I don't speak Spanish and I don't speak Italian, sadly. This is myself and a colleague called Mario, one of the peer interviewers at a farm near Caserta, which is near Napoli in Italy. I'll provide a little bit of background about the sites. There were 2 sites in Italy, and there are 2 producer associations. These are quite important to the whole thing. They manage contracts and they provide agronomic advice. In Italy, they grow a dark type of tobacco, which is all handpicked, which is quite incredible, and it's used in these kind of what I would call a small cigar, called Toscanos. The average farm size there was about 7.3 hectares, which is about 10 football pitches. But they also grow olives and wheat and vegetables. Very few tobacco farmers were only tobacco farms. In Extremadura, there were 3 producer associations, and they do the same kind of stuff, but they also have these communal drying facilities to dry the tobacco before sale. And there they grow Virginia, which is probably the most widespread form of tobacco, and that can be mechanically harvested. So and it's used in regular cigarettes. So you see that the mean farm size is much bigger. It's about 70 football pitches each farmer had, and they grow corn and peaches and things like that, and the peppers that go into pimenton de la vera, which is like a posh paprika you might have had. So what did we find when we spoke to these guys? They complained about price quite a lot, which they claimed had stagnated. And in Spain, in fact, they only really survive on subsidies. And the latest data that we could find kind of corroborated that story. The price that is paid to the tobacco producers has been stagnant for a long time. Everyone spoke about climate change and extreme weather, which is very interesting. They like to complain about pest control products being lost, which in my experience is a very general thing farmers complain about. Due to European Union rules, a lot of chemicals are being taken off the market, essentially. They found it more and more difficult to find workers, and they had a variety of succession problems, passing on the farm, that kind of thing. This farmer was speaking about how in the past it was far wetter, and it's more and more dry, relating to climate change. There was a lot of stigma around being a tobacco producer, even though they had a very strong identity as tobacco producers. I am a tobacco farmer, said one. I am the son of a tobacco farmer. I am the grandson of a tobacco farmer, which is quite interesting from a social science perspective. But they had a strong desire to continue growing it as well. And most said that the final use of tobacco wasn't that important to them, despite the fact that when we told them about plant molecular farming and explained it, they were very supportive of it. They thought it was a good idea. One Spanish farmer said, "I don't know how to describe it. It would be like meeting the Messiah if tobacco could save lives." And interestingly, most were fine with GM crops if it was for this purpose, for molecular farming. Some, not all, but some wouldn't eat GM food. They were against it. An Italian farmer said, "I mean, as a grower, we have always been against GM food. But in this case, you understand the purpose is different." So we hypothesized that purpose was key to acceptability. And they also thought that plant molecular farming could improve their image in the public eye because they'd be producing medicines and things. So this is quite interesting when you're thinking about a speculative technology, that something is more acceptable than something else, and what are the reasons? In terms of sort of agricultural system and how it would work, then there's a high level of trust in the existing arrangements, and particularly with their producer associations who manage a lot of things for them. So one Italian farmer said, "It is a good association. They're interested in tobacco. We believe in tobacco. We're pleased. We can hope to continue with this organization." And this leads us to a few conclusions later on. They also were concerned about what kinds of things that New Cotiana's tobacco might mean for infrastructure. Do they need new tools, new equipment? And for agronomy, so how to manage this new crop that they've never seen or never practiced growing. So the main concern, said one Italian technician, was that we should see which kind of plant they are going to create, the development, maturation, how long the growing cycle is. So they were quite keen to get their hands on it. But the final thing is that, of course, tobacco is bought from these associations, from these farmers, by very large companies with a lot of power historically to dictate various kinds of things. And they've historically been opposed to genetic modification of any kind. A Spanish policy expert told me that until now, they've not been in favor of this kind of thing. When there were talks about genetically modifying tobacco farming, they never agreed. So their involvement in New Cotiana, because one of our partners was one of these big tobacco organizations, they didn't take any money. They just were able to sit in on the meetings and things. It's not a coincidence. There's a reason. So he thought that they were kind of opening up to this kind of thing. Yeah, and big tobacco have often invoked rural livelihoods as a reason to avoid regulation. So they say, don't regulate tobacco because you'll have this knock-on effect on all these producers and things. So, taking all that, what we said would be key to scaling up is that new end use for tobacco is better than just substituting it with other crops, as the EU at that time was trying to do, because farmers wanted to keep growing tobacco and they have a lot of expertise in it. So partial substitution makes use of their knowledge, basically. The higher premium that you could potentially get for plant molecular farming products could be offset by different practices, but then this equation is quite difficult to determine because one of the aims of New Cotiana was to reduce flowering, which was something they had to control either manually by clipping the flowers or chemically. So that could potentially reduce costs for them. So there was this kind of equation we had to figure out about what the eventual price would be for them. And to be honest, because it was so early in the project, we didn't have answers for them on that. We felt that producer associations would be key to that process because they can help lease equipment, provide loans, and arrange the contractual agreements between the farmers and then the buyers, who would probably be some kind of pharmaceutical companies. There's also a big association of associations called UNITAB, and we thought that would be the kind of organization that would need to be involved in something like this to promote acceptability. So the strengths of the approach in this case are that there have been calls for farmer voices in discussions about biotechnology, but there haven't always been farmer voices in that discussion. So this is one way of capturing those. It makes plant science a little bit less remote for its potential end users like farmers. And I think it's quite an interesting process as a researcher to go out and do this kind of thing, and you get quite interesting outputs as well. The stuff about climate change is really interesting, sort of people who experience it on the ground, and stigma. All this stuff is very interesting from a sociological perspective. That said, there are a few limitations. You're kind of at the mercy of who you can find to help you do the research. If you need help, which we certainly did, we got lucky. I would always recommend taking a lot of gifts out with you as well, because you'll be in weird debts to people who are just helping you. One participant described the project as air, because it was so speculative at that time, and people like to experiment with new technologies rather than just talking about them. There's also difficulty in influencing the plant science at that stage of the project, and limited impact. The limited impact you have on the development of the technology, and that's something we're still kind of grappling with. And I appreciate that this is probably a different field entirely from what most of you are concentrating on, but I hope there's some cross-fertilization of knowledge, and if you have any questions, you can just ask.